Our first speaker is Chris Exley, who is a biologist specializing in bioinorganic chemistry. He received his PhD in the ecotoxicology of aluminium from University of Stirling in Scotland and joined Keele University in England in 1992 to establish the Birchall Center with J.D. Birchall. Dr. Exley was awarded a Royal Society University Research Fellowship in 1994 and elected Fellow of the Royal Society of Biology in 2015. Recognized as a world leader in aluminium research, he has over 200 peer-reviewed publications in the medical literature. His new book, Imagine You Are an Aluminium Atom, will be published later this year.
Oh, hello. So my name is Chris Exley, and I'm talking to you from over here in the United Kingdom. I'm at Keele University, and all the details, as you can see, here on the slide. I really want to thank Barbara Lowe Fisher, and primarily, of course, for the original invitation to come to the US and give my talk at this, very important meeting. So now I'm going to go into my sort of slightly grumpy mode because I don't actually do these types of presentations. This is probably the only Zoom or whatever you call it presentation I've ever made, and it's possibly going to be the last one. And there are a number of reasons why I don't do it. One is I don't agree with anything to do with the new normal. In other words, when I like to talk to people, I like to be able to see them there and then in front of me. I like to have a live audience. I like to see the whites of their eyes, and I like to interact with you. And many of you who know me will have that experience of me. So being sat here, unable to hardly move and talk to no one is a rather unusual experience for me, but I'll do my best. And, the other thing I have to point out is that those of you who saw me last giving a live talk, which I think was back in February now, will remember that I was doing a good impression of Shane McGowan from the Pogues. And I still am giving that impression because, as you might see, I still haven't managed to find a dentist in the world of COVID who can fix my broken crown. So don't expect any smiling from me. You're not going to get any smiling in this presentation. Maybe just a small one. So thank you, Barbara. And this is especially for you. Now, look, the title of my talk is, this is a life's work. And if you want to understand about it, go onto my website and it's all there for you in different formats. You can read about it. You can listen to it on videos. You can listen to it in various interviews that I've given. So today, really, I'm much more interested in focusing the message that I want to give to you all. I want it to be a very specific message, something for you to carry and take away from this meeting. It's not a fun message. So I want the message to be essentially what I'm about to show you on the next slide. And that is that, while many of us are believing or not that we're living through some sort of pandemic at the moment, in my opinion, there is a true pandemic that devastates human lives each and every day. And that pandemic is our continuous and burgeoning exposure to aluminium. Aluminium is the English pronunciation, as I know you all know. So I'm going to stick to that because I will just get tongue-tied if I try with aluminum too often. So that is the message I want to put across. And let's just develop that message and why I feel so strongly now, after, my maths is not great, but it's probably 35 years of research in this field. If we look at everything I show you, by the way, there are links available to published research. So don't worry too much about having to digest those details of the slides at this moment in time. I'd rather, in many respects, I often tell many of my students when I'm lecturing to them, I say, just listen to me. This is a one-off on this occasion. But what this particular slide is telling us, it's what I call the biogeochemical cycle of aluminium. But the message is that for all of evolution of life on Earth, nature has recycled aluminium, the third most abundant element of the Earth's crust after oxygen and silicon, has recycled it with such efficiency that no biologically available aluminium enters the living cycle. So for 99.9999999% time of life on Earth, life did not encounter significant amounts of biologically available aluminium until man intervened. And that's what I've got here under human intervention. So let's look at that in more detail. I call that period in time when the world changed, when the Earth changed, when biologically available aluminium entered the living cycle. I call it the aluminium age. And you'll see from the slide, it's just a tiny fraction of a period of time. And it corresponds to primarily when man learned how to take aluminium ores, inert aluminium ores, out of the Earth's crust, and from them make aluminium metal, and from the metal make aluminium salts, and from the combination of having now the metal and the salts, we had the advent of the aluminium age. And if I was to simply list to you the number of ways in which we use aluminium in our everyday lives now, it would take a minimum of 24 hours of continuous listing. So the aluminium age is wholly and completely upon us, primarily due to the extraction of aluminium ores from the Earth, but also some indirect ways that you will probably have heard of. One being acid rain, the acidification of environments that would normally not be acidic because of rain, acidic rain falling upon them. The acid rain coming, of course, from the burning of fossil fuels. So man's burning of fossil fuels is releasing acidity into the atmosphere that would otherwise be there, falling onto the land, polluting the soils and our lakes, killing fish and trees. Acid rain. And the other way, which is again perhaps a modern, relatively modern phenomenon, is intensive agriculture. So when you grow crops intensively on soils, you will acidify those soils over time. And intensive agriculture has now resulted in more than 30% of the world's agricultural soils being acidified, such that they release aluminium from what would otherwise be completely inert stores. So there we have it, the aluminium age. And as I've always nearly briefly mentioned about this, the consequences of this can be seen in the evolutionary consequences. I was asked in 2009 to write something about this subject as part of the celebrations of 200 years since the birth of Darwin, or 150 years since the publication of The Origin of Species, to explain essentially how aluminium and silicon do not have biological functions, even though they are the second and third most abundant elements of the Earth's crust. But again, the main take-home message, which is shown here to a certain extent in my version of Darwin's Tree of Life, is that when something new enters the biochemical evolution, the evolutionary process. And that is an example of that is aluminium in only the last 100 to 150 years. It is inevitable that it has a negative impact in the first instance. So there are many examples of this throughout the natural selection of the elements, throughout evolution and biochemical evolution. We've seen where toxins become essential elements, toxins like oxygen becoming essential for oxidative metabolizers like us, toxins like calcium becoming essential in signaling, etc., and bone formation. But aluminium is only just participating in this process, and everything we will see from aluminium for now and for quite some time to come will be negative. Biologically available aluminium can only be negative. So how are we as human beings exposed to aluminium? So this is my, again, schematic of human exposure to aluminium. It is complicated. The figure suggests that it is, and that is the case. And the truth is that there is a dearth of high-quality research into human exposure to aluminium. Not because of a lack of wanting to do it, but simply because of a lack of funding. And the truth is that we only really see, the old cliche, we only know about the tip of this particular iceberg. Don't get me wrong, what you will hear, as we are hearing today about COVID, you will hear convenient truths being spoken all the time about human exposure to aluminium. These convenient truths are perpetrated by the aluminium industry, although largely through the governments of our respective countries. Again, to use some of the vernacular of today, there are no fact checkers looking at the lies that are being perpetrated by governments on human exposure to aluminium. Well, there might be one or two, and I might be one of them. But who's listening to me, I wonder. So what does the aluminium age actually look like? Oh, here it is, to a certain extent. Here is the tip of the iceberg. Here is what we probably begin to understand about what it's like to live in the aluminium age today. I'm guessing that you can recognize a few of these conditions, and of course you can. They are some of the top 10, top 20 chronic human diseases, and aluminium has been implicated in all of them. You've got to ask, these are top 10, top 20 chronic human diseases. We do not have or know of a cause of any of them. There is certainly no cures for any of them. There is hardly any effective treatments for any of them, and there's virtually no way of preventing them. How is this possible? To me, it's only possible because of the paradigm being that there's only money in disease. Until there is money in health, that won't change. But there is money in health, so we need to make a paradigm shift such that we can make health the major issue and not disease. And then medicine will have a vital role to play within that. At the moment, medicine is just a way of making money. And it's very sad. I want to focus a little bit on the infant. And that's primarily because we all start off in this particular guise. And when we are born, our nutrition primarily comes either from our mother's milk, and that can contain some aluminium. So that is a possible way in which you might be exposed to aluminium. The breast milk, generally, depending on the mother, will not have very high levels of aluminium. The same cannot be said of infant formulas. Infant formulas are a scandal. It is quite unbelievable to me that we feed our infants with products which are so heavily contaminated with aluminium. We would not allow that level of contamination in anything else, in our water supplies or anything. But in infant formulas, it's apparently perfectly okay. It is a scandal. And of course, you're talking about how infants receive in the first days, weeks, months of life are exposed to aluminium. Well, another way, of course, which many of you are aware of, will be vaccinations, aluminium adjuvants in vaccines. Another very high exposure, particularly in infants and particularly in, of course, newborns in the first months of their lives. So how is an infant placed to deal with such a high exposure to aluminium? Well, the answer is very poorly, because as I argued a few years ago at an Autism 1 meeting, the infant is in fact the perfect model of human intoxication. First of all, because of the ways in which we've just discussed, in ways in which it is exposed to aluminium. But then you have to think about, well, the infant's skin is underdeveloped. Anything applied to the skin of an infant it's more likely to let aluminium through than when the infant is older. If anything that goes into the gut of an infant, the gut is underdeveloped. Aluminium passes more easily from the gut into the bloodstream. When aluminium is in the bloodstream, the kidneys that are normally filtering aluminium and helping to excrete it by the urine are also underdeveloped, and so less aluminium is removed from the blood. Less aluminium is removed from the blood, meaning that when it comes across the blood-brain barrier of the infant, which is significantly underdeveloped in the first 6 to 12 months of life, more aluminium can get into brain tissue. Now, finally, add to that infants do not perspire very much. They produce very little sweat. And sweating along with the urine is one of the main ways in which aluminium leaves the body. So the infant, it almost makes me feel ill to say this, is this almost perfect model of aluminium intoxication. We know all of these things to be true, and yet we do absolutely nothing about it, at least at a regulatory level. I'm having a little drink of a silicate-rich mineral water. Some of you will know the significance of that. However, yes, human exposure, the infant is very important. We're also finding out that actually exposure to aluminium starts at conception. Aluminium, we find it in, for example, human sperm. The image shown on the right, the orange fluorescent image, shows aluminium in human sperm. We know that the aluminium is in ova, in the eggs. We know that it's in the mother. The mother has a body burden of aluminium, and we can see that in the image on the left in the placenta. Perhaps, again, even more worrying, if one needs to worry even more, is that some recent research, and there is some good research getting done, which is fantastic. You'll see at the top of the slide, Toisi et al. Looked at metals in serum and looked at congenital malformations in infants. So, they looked at metals in the serum of pregnant mothers, and then they looked at congenital malformations of the nervous system in the infants that were born to those mothers. And they looked at a great range of metals, and the only metal that showed any relationship was aluminium. In other words, a strong relationship between aluminium in your blood and congenital malformations of the nervous system. So the nervous system. This is where I want to just concentrate the rest of the talk. Aluminium in the human brain. And, the reason for that being that perhaps we know more about aluminium in human brain tissue than we do in almost any other part of the human body. And I think it's probably also what we should be most concerned about or even scared about. And let's sort of see why. Again, aluminium in human brain tissue, how much is too much? Why did I even have to ask this question in a publication? How much is too much? Do we have to ask the same question again about any other neurotoxin in brain tissue? Do we think, well, that's perfectly all right as long as there's only this amount of a known neurotoxin in brain tissue? I don't think we do. Aluminium in brain tissue is only neurotoxic. That's all it can be. It has no function, no positive. It can only be neurotoxic. So all aluminium in brain tissue is bad news. Of course, that's not exactly how science works. So you've got to convince those naysayers, and of course there are many of them, who for some reason or other do not think that aluminium in human brain tissue is an issue. So what we've done, and this is actually done, research published very recently, just this year, we've looked at the aluminium content in human brain tissue in individuals with no neurodegeneration, neurodevelopmental disease at all. And we compared the amount we found in those with brain tissue from people with Alzheimer's disease, multiple sclerosis, and autism. And this is what we found. And I know you know this already, because most of you probably, unlike the vast general population, are tuned into this subject area to a certain extent. But we can see very clearly from the graph that the amount of aluminium in human brain tissue in the absence of any neurodegenerative or neurodevelopmental disease is very low. It's still too high as far as I'm concerned, but it's very low. And look at how much lower it is, how much statistically significantly lower it is than sporadic Alzheimer's disease, the main form of Alzheimer's disease, familial Alzheimer's disease, we'll come to a little bit more about that in a minute, autism, and multiple sclerosis. So this paper, published in Scientific Reports, it's the type of paper, again, that 20 years ago would have been published in Science or Nature. And by now, I think we would have addressed the problem of aluminium and human exposure to aluminium. But we are fighting against a massive political propaganda organization that does not want the truth about aluminium and aluminium in brain tissue to be known. And again, this is the sort of paper that should have been on the news at 10. The data should be there. They should be in your local newspaper. But you know what? You didn't hear this wherever you are in the world. You did not hear about this on your news channel. You did not read about it in any of your major newspapers. This is the power of the aluminium industry and those that work with it through governments to suppress research. So there is too much aluminium in brain tissue, in Alzheimer's disease, in autism, and in multiple sclerosis. When we published our first research on aluminium in familial Alzheimer's disease, and we showed that the levels were higher than almost anything we'd measured before, again, I thought this was like a landmark. This was going to change the world. This was going to tell us that there has to be and must be a very significant contribution of aluminium to the etiology of Alzheimer's disease. But no, the paper just disappeared into obscurity. The research essentially disappeared into obscurity. So we did it again. This time we found another cohort of individuals with familial Alzheimer's disease in Colombia, in South America, on the other side of the world from the London cohort. And we measured the aluminium content of their brain tissue, and we found equally high amounts of aluminium in their brain tissue too. And of course, just to prove the point, we can compare them with control brain tissues where there is no neurodegenerative disease at all, and you can see that the amount is massively, statistically significantly higher in familial Alzheimer's disease. So the evidence is mounting up. This paper was published in the Journal of Alzheimer's Disease. The journal itself released a press release, put out a press release in which the editor, George Perry, one of the world's leading researchers in Alzheimer's disease, called it a landmark study. It didn't make any press whatsoever, even with the support of one of the world's leading researchers on Alzheimer's disease. When they want to prevent something from being known, they prevent it from being known. And just to add to this, when we got the tissues from multiple sclerosis, from the multiple sclerosis brain bank, at the time they also sent us what they thought would be good control tissues. At the time, we weren't happy that they were good control tissues because while they were from individuals that didn't have multiple sclerosis, many of the individuals in the control group had died of quite traumatic diseases, and many of them, a large proportion, from cancer. However, we measured these brain tissues, and we made a comparison between them as what we considered to be not a perfect control versus multiple sclerosis. And once again, there was absolutely no doubting that the amount of aluminium in brain tissue in multiple sclerosis is higher than an age and gender matched group that did not have multiple sclerosis or any other neurodegenerative or neurodevelopmental disease, but did die of some other quite catastrophic conditions. How much aluminium in brain tissue is too much? This is too much. As far as I'm concerned, this is too much. So there's too much aluminium. But you know what? Quantitative analysis, the internet trolls that are out there that are paid by the industry to try to dismiss research, they have done their best to look at quantitative analyses and to try to destroy them in their non-reviewed internet blogs, the likes of Gorski and others, terrible individuals. But what about if we not combine the quantitative analysis with imaging of aluminium in brain tissue? What does it look like in brain tissue? And one of the big advances we've made over the last 5 years or so was to develop a method which unequivocally shows the presence of aluminium in tissue, and in our cases, human brain tissue. And it uses a method known as fluorescence microscopy and a molecule known as lumogallium, which binds with aluminium to produce a characteristic fluorescence. This fluorescence is absolutely selective for aluminium. While this molecule will bind other metals, they do not produce fluorescence. So we have a method now where we can image and view aluminium in human brain tissue. And for me, this is where things really began to get very serious indeed. Let's start with, now these images, from all images I show you from now on, have been produced by my wonderful research fellow, Dr. Matthew Mould, who is just becoming a real genius with the microscope. So this is some of the first images of aluminium in human brain tissue that really started to make me really worry about aluminium in human brain tissue, because these are images taken from an individual who died with autism. And we were able to look at 10 individuals using fluorescence microscopy, and we found similar images to these in all 10 individuals. What you're seeing on this image here at the moment, if you look particularly in the insert at the top left, these are lymphocytes. These are white blood cells, and inside them you can see little specks of white, yellow, orange light. They are, what you're seeing is aluminium, probably inside a vesicle of some sort within the cell, within the lymphocyte. And if you imagine it like a little fluorescent light bulb, it lights up with real intensity the more aluminium is present, and indeed it can then light up the area around it in the same way as a fluorescent light bulb would do in your room. So what we're seeing here in this image were cells derived from the body, lymphocytes, crossing the meninges and presumably going into brain tissue in someone with autism, diagnosis of autism. This is significant, and we'll talk about it a little later. If we also look at another image taken from autism brain tissue, now looking into the brain tissue itself, here we see what looks like perhaps an area of high intensity showing aluminium in possibly in a damaged tissue within the brain itself, maybe damaged neuronal tissue. And around it, we see the glial cells. They're like the lymphocytes of the brain, the housekeeping cells of the brain. And they seem to be almost, well, having migrated perhaps towards that site of damage. You're almost thinking, here we've got some damage induced by aluminium in autism brain tissue, in this case, a 15-year-old an adolescent male. And of course, when you get some sort of damaged tissue in the brain will send out signals asking for help. And usually the help comes in the form of the glial cells, who come to try to help out in many different ways. But if those glial cells are also loaded up with aluminium, they can't help. In fact, all they can do is exacerbate things and make things worse. And I'm sure that that is what we're seeing here in this tissue. We can see something similar to this in epilepsy tissue. This is a case of late onset adult epilepsy, where we found a lot of aluminium in both the hippocampus of this individual and in the frontal cortex. And this is a part of the frontal cortex, again, where you're seeing neuronal debris. You're seeing what looks like an area highly fluorescent for aluminium, extracellular debris that's giving a strong aluminium positive reaction, but close to it are glial cells. And look at the insert, look at the glial cell full of little specks of aluminium, probably inside vesicles. Very similar to what we saw, of course, in autism. And of course, in many of the autism cases we looked at, many of those also had epilepsy. So moving on, look, familial Alzheimer's disease is the early onset form of the disease, and it's in individuals may get Alzheimer's in their late 30s, 40s, and 50s. And the genetic mutation, which is implicated as a predisposition to this, results in a larger production of a protein called amyloid beta in the brain tissue. And amyloid beta is considered by most Alzheimer's researchers to be the #1 primary etiological factor in Alzheimer's. So what we showed in our Colombian familial Alzheimer's disease brain tissues, we were able to do some fluorescent microscopy to look for aluminium. We were able to do some complementary fluorescent microscopy to look for amyloid and seeing whether or not there was any relationship between the two. And in fact, the relationship was remarkable. The co-localization of aluminium and amyloid beta in these brain tissues was in every single case that we looked at, and multiple times throughout all regions almost of the brain. So what we see in the image here, in A, we see aluminium, the orange is aluminium. In B, we're using a different technique, and the fluorescent green is amyloid beta. And in C, you see their co-location. D is a method using something called Congo red, which simply tells us that we're right, that the fluorescent marker used in B is picking up amyloid. Another example, again, from familial Alzheimer's disease. We look at B, we can see a round plaque-like structure giving a very positive signal for aluminium. When we take the same tissue, but we're looking for amyloid beta only, we see a lovely, well, lovely to look at arrangement of amyloid and amyloid fibrils and threads forming a sort of senile plaque. And when we look at them together, we see this remarkable image of co-localization with all these amyloid threads around the aluminium. And this was something we saw over and over again in familial Alzheimer's disease. The other thing we saw on a regular basis in familial Alzheimer's disease was the presence of both aluminium, as seen here in B, and amyloid beta, seen in C, together in the blood vessels. And this is part of an Alzheimer's called conjunctival amyloid angiopathy, where not only do you get senile plaques, but you can also get the presence of amyloid in the blood vessels in the blood-brain barrier. And here we see them co-located, again, aluminium and beta amyloid. Okay, so this is a little bit of a heads up, because the study that we have ongoing at the moment and was completely brought to stop by COVID is Parkinson's disease. I know many of you will be interested in Parkinson's disease. So we've released a little bit of information early, and this paper is about to be published. But what we're seeing here, and essentially it's easy just to look at images in A and C and then in D. Now we're looking for the presence of aluminium. We can see it here in A. In C, we're now actually looking for another one of the main etiological factors, both in Alzheimer's disease and Parkinson's disease, which is called tau. And we're seeing tau here as indicated by the asterisks. And we're seeing them perfectly co-located, we can see in D. So we're seeing aluminium and tau together in brain tissue in Parkinson's disease. I'm afraid you're going to have to wait a bit longer for the rest of the data from Parkinson's. It's going to be another 3 to 6 months before that is ready for submission for publication. We are getting back on with that now. And as you can see from the early work, it's going to be exciting. Okay, so this is my last slide, real slide. And the purpose of it is to make you think a little bit about how my mind changed, for example, with respect to whether aluminium could have a role in infant deaths following vaccination, or aluminium could have a role in infant disease like autism. So on the left, we're seeing something called a T helper 1 cell. This is the type of cell that infiltrates a vaccine injection site. And what you're being shown on the left is this cell is got discrete packages of aluminium in its cytoplasm. So it goes to the injection site and it picks up the aluminium adjuvant at the injection site and encloses it in membranes. And these are cells that can move about the body. So they can go off to the lymph nodes, or they can go to other organs. So they can take with them a significant cargo of toxic aluminium. If we look at the one on the right, this goes back to the picture, the first image of aluminium fluorescence that I showed you in the autism brain tissue. It's a lymphocyte. Look at it. It's also inside got these discrete packages of aluminium. And this lymphocyte was crossing the blood-brain barrier of an individual who died with the diagnosis of autism. I asked the question when we published our research on autism, how is it possible a 15-year-old boy had some of the highest levels of aluminium in their brain tissue that we have yet measured. And for me, the only answer to that, well, if I go backwards one step, up until I saw these images, my understanding was that we accumulated aluminium in our brain tissue through our normal living over decades. And that is why perhaps we see things like Alzheimer's and others in later in life, not earlier in life. But when I saw these images, I realized there is a mechanism whereby aluminium can enter brain tissue in very significant amounts within relatively short periods of time. In fact, it's not impossible that aluminium from the injection site of a vaccine could be within the brain tissue within hours, days afterwards, and is not impossible that it could cause an encephalopathy within that tissue. And that encephalopathy could then result in either permanent damage or indeed death. This is what changed my mind. And this was just a few years ago, before which I didn't see a way in which aluminium could be involved in a disease like autism, or I did not see a way in which an aluminium adjuvant could produce or could be responsible for the death of an infant within the first few days of their life. There is a mechanism now. We really do need to be very careful about this. It's a very difficult question to answer whether or not a child in the first few days, weeks, months of their life should receive a vaccine containing an aluminium adjuvant. I'm glad that I personally don't have to make that decision, because I would probably go against it and not accept that particular vaccine now, simply because of what I know. We can make vaccines much safer than they are today, and part of the research in my group is all about vaccine safety. So a little bit of advertising to finish my talk. This is my forthcoming and essentially only book. It's a no-holds-barred account of the last 30-odd years of desperately trying to bring the story of aluminium to the world. And I hope if you read it, you will enjoy it, but most of all, you will understand why I've done it, why it's necessary, and why we need things to change. Thank you very much.